everyone, welcome to this class, and today we'll talk about basic concept behind finding of gene order in 3-point map, and this is very important for the CSIR examination, and this are kind of questions usually like it comes actually in CSI examination. So this is very important topic for you, and if you are new to the channel, make sure to subscribe my channel and press the notification bell and like the video and share the video. So this is a quick like conceptual class for you, where you will learn how to find the gene order perfectly. So there are so many ways you might aware of, but what I will show you today, and this is like more risk free method, where you will find the gene order correctly every time, every time. So you know about the 3-point cross. So 3-point cross के बारे में अगर आप लोग जानते हैं, so this is very good. And if you don't know about 3-point cross, so there is one or two videos are there where I explain the 3-point cross to you. So make sure you watch that video and you learn this 3-point cross, 3-point mapping, or 3-point cross, anything you can say. So in the 3-point cross, you will get 3 genes, okay, for 3 particular locus. For example, we are considering one specific genotype, and this genotype can be written like this. And always in gene mapping, you should consider test cross if you want to find out gene map, or you want to find out distances between genes. So this is one test cross. You can see, you can check easily. This is a test cross. So like from this cross, you will get 8 different types of gametes, because this is one tri-hybrid genotype, and 3 loci are hybrid loci. So you will get 8 different gametes from this individual, and that's why 8 different genotypes. And what are those genotypes? A, B, C from this parent, and the same gamete from this parent, always the same, that is small a, small b, small c. So this is one genotype where there is no crossing over, and there like crossing over did not take place in this particular gamete, in this particular genotype. So this is one genotype where crossing over did not take place. So another genotype is this one where crossing over did not take place. So these two are non-crossover types. You know that. I'm like telling you quickly. And in other cases, so there can be a possibility of single crossover. There can be possibility of double crossover. Okay. So when single crossover happen, okay, then what will happen? So in single crossover, like this kind of thing will happen. A, B, C, capital A, B, C, small a, B, C. In this genotype, single crossover between A and B, that means crossover will take place here. So we will get genotype capital A, small b, small c, capital A, small b, small c, and in the denominator it will be small a, small b, small c always. And other like genotype would be small a, capital B, capital C, and in the denominator small a, small b, small c. This is the single crossover, that means SCO, single crossover between which 2 genes? A and B. Okay, so this is 1, this is 2, this is 3, this is 4, and similarly there can be crossover between B and C also. So if the crossover happens between B and C, what we will get? Capital A, capital B, small c, and the denominator same, small a, small b, small c, and also small a, small b, capital C, and denominator small a small b small c. These two are single crossover between b and c, as we have like observed. So between b and c, and apart from these six types, there will be one pair of double crossover genotypes. So the double crossover genotypes will get this one. There will be two crossover and we'll get capital A small b capital C, denominator small a small b small c, and another one small a capital B small c, small a capital B small c, denominator small a small b small c. So this 7 and 8 1 this, so total 8 genotypes we'll get. So these two are very important. These are double crossovers, double crossover type of genotypes. Okay, so you can easily understand. Like, I hope you understood this part clearly. So now for to find out the gene order correctly, so we need two types, like two pairs of genotype from these 8 genotypes. Okay, so what are those types? So we have to consider non-crossover types. That means this one and this one, and these are non-crossover types, NCO non-crossover types, and another one double crossover. That means capital A small b capital C and small a capital B small c. But here we know that what is the correct gene order. The correct gene order is A, B, C. This is the correct gene order. Or we can say C, B, A. Same thing. Okay, the A, B, C and C, B, A is the same thing. So the most important thing is the middle gene. Here the most important thing is the middle gene, because the middle gene is constant in this order. So if you can find out the middle gene, so then you can easily find out the gene order. Okay, so how can we do that? So let's say we don't know about the correct gene order, that is A, B, C or C, B, A. Let's say we don't know this, but we know the double crossover types, double crossover genotypes, and also we know the non-crossover types where crossing over did not take place. So these two are double crossover types and these two are non-crossover types. But let's say we don't know the original gene order, so we need to find out these two. This gene order we need to find out. So how can we do that? So just in this scenario, in this case, you need to take the numerator only. Okay, from these two double crossover genotypes, you need to take the numerators only, because the denominator comes from different parent, and the crossing over you can see the crossing over has taken place in this genotype, not in this. Okay, so we need to consider only the numerator. So let's write the numerator, capital A small b capital C, and in the denominator small a capital B small c, because these two numerator came from a single parent. Okay, so this is a double crossover cumulatively. We wrote this down in a cumulative manner, and in this same fashion, what we can write the non-crossover types. So we can write cumulatively the non-crossover types as capital A capital B capital C, the numerators only, and small a small b small c. But let's say we don't know the correct gene order yet. So just think carefully and see carefully. In this double crossover types, okay, if we make another double crossover, okay, another double crossover event in this double crossover, in the double crossover, I'm telling you again, in the double crossover, if we make another round of double crossover, so then what we will get? We will get the original non-crossover types. Okay, if we make a double crossover in the double crossover types, then automatically we will get this non-crossover types. So this is the key, this is the main factor here. Okay, if we like in the question, I will show you another one question, then you can understand this thing clearly, even clearly you can understand. So like this way you will easily get an idea that what will be the correct gene order. If the double crossover, if we do a double crossover in the double crossover, and then you will get the non-crossover types perfectly, then you can say that this gene order is correct. Which gene order? This gene order, A, B, C is correct. But if you don't get the correct match, okay, if you make a double crossover in the double crossover and you did not get the non-crossover types, so then you cannot say that this is the correct gene order, because okay, so you consider this gene order as correct. Previously you considered this gene order as a correct gene order. Then you made the double crossover. Okay, and if you get the wrong match, then you cannot say this is the correct gene order. So what you can do in that case, if you did not get a match, so then in this double crossover genotype, you just alter this middle gene portion. Okay, you replace this position. You replace this position with either C or A. That means you can do this. Okay, just make sure the genotype is same, remains same, but you just change the position. Okay, you just change the position like this. B, small b, capital B. Okay, we just change the position. Capital A, small a, and capital C, small c. Just like this. Just we made the change. Now again you will try to match like this way. Now again you will try to match like this way. Small b, capital A, so small b, small a, capital C, capital B, capital A, small c. And if you read like this, so you can, what you will read? What you will read? You will read like this. From this one, I am changing the color so you can understand. If you read like this way, small b, small a, capital C, so you can read like this way, small a, small b, capital C, right? From this line, and from this line you will read like this, capital A, capital B, small c. But is it the correct match? So like, did you get the correct match? No. The correct match is this one, capital A, capital B, capital C on one side, and small a, small b, small c on another side. But you got small a, small b, capital C on one side, and capital A, capital B, small c on another side. So you can say confidently that this BAC gene order cannot be correct. Okay, and similarly you just change the position of which position? The middle gene position, the middle locus. Just you change the middle locus and try to match. and in this way you will get 100% match. 100% match if you positioned the correct gene in the middle place. Okay, you can, if you position the correct gene, okay, and follow this method, follow this way, so you will get the 100% correct match. So now we will see from an example, and this example is one question that came in the CSIR examination. Okay, I just took that question. So in Drosophila bar I, that is capital B, this is dominant mutation, while miniature wing and yellow body color, that is small y, are recessive mutations. So heterozygous females for these mutations were crossed, okay, to the normal-eyed miniature winged. Okay, heterozygous females for these mutations. So heterozygous females means, we have to assume, for if you don't know the correct gene order, but you have to assume something, and you have to start from somewhere, right? So heterozygous females from these mutations were crossed to normal-eyed miniature winged and yellow body colored males. Okay, so let's do the cross, because cross is important. So what will cross? Heterozygous females for this mutation, this bar I and miniature wing and yellow body. Bar I is dominant. Okay, bar I is dominant, but normal I is recessive. Miniature wing is dominant mutation, while miniature wing and yellow body are recessive. That means miniature wing is recessive, yellow body color is recessive. Okay, so we will do this cross, B plus M plus Y plus, just we assume. Okay, we don't know that this is the correct gene order or not, we don't know, but we have to check. Okay, just we have to check. So how can we proceed this one? So just from this table, you need to find out first, like which pair of genotypes are if we write here genotype, that is also correct. If we write here genotype, that is also correct. Like here they wrote phenotype, like we took that. Okay, so we have to find out what are the non-crossover pairs. So for this you need to find out the highest number. Highest number is 185, that is B plus MY, and the complementary allelic conformation is the partner of this genotype. Okay, we need two, right? So what will be the partner of this genotype? The complementary allelic configuration. So what can be that? B M plus Y plus B M plus Y plus. So where it is? B M plus Y plus, this one, 165. So 185, 165. So these two are the parental combination, and we need to match, right? With what? We need to match with the double crossover types, and we will make double crossover in the double crossover and try to match with this non-crossover. This is the non-crossover. We will try to match with this non-crossover. So let's find out the double crossover. So the lowest number, we need to find out the lowest number. What is the lowest number? 20. And what is the genotype? Genotype is BMY. And then what will be the partner of this genotype? The partner of this genotype must be B plus M plus Y plus, isn't it? So this will be what? This one 30, not 25. So don't make this mistake that the lowest two, no, not always the lowest two. The lowest and its partner, and in this case the partner is B plus M plus Y plus, and this is 30, not not 25, this is 30, and the partner is 20. Okay, so now we have to do our method. So our method is, we have to make the double crossover in this double crossover combination. So this is double crossover, and we need to do double crossover. B m plus y, B m plus y, and is this like any combination in a numerator or denominator which is b m plus y? B m plus y, no. This is b plus my and b m plus y plus. So no b m plus y is present. Okay, so for like with this information we can say that b m y gene order cannot be possible, right? So b m y cannot be possible. Then let's just change the middle portion, like middle locus. Change the middle locus with the neighboring gene. So let's follow the option here. Option says B in the middle. B in the middle, but make sure you don't change the like allele configuration. So allele configuration is same, but just you change the position. Okay, you change I just changed the position. Now let's do it again. Now m b plus y, and we have to read it like this way, right? B plus m y. B plus m y is there, yes. The numerator has been matched. So now another one, m plus b y plus. So we have to read it like this way, b m plus y plus, and it is also a match. It is also a match, right? So we can say that this m b y, this gene order is correct. Okay, so we have conclusive evidence that this gene order is correct, MBY is correct. Okay, so like you can easily see that in this method, so there is no chance of error. There is no chance of error. You will get always the correct option. You can always find out the correct option. Okay, now for the gene distance portion, I hope you can solve that. And I also took that class where I explained that three point mapping and this cross concept regarding this cross. So I hope you can easily solve this, right? So for this class, I hope I made you understand how to do how to find out the correct gene order without any error. And if you follow this method, I can assure you, you will always get the correct match. You will always get the correct option. You can always find out the correct option. So just you have to trust my method. That's all. Okay. So I hope you like the class, and make sure you watch the other videos which are also there in the playlist, and the classes regarding 3-point mapping, okay, and the probability, and some questions we have solved with the help of this method, with the context of this 3-point mapping. We have also covered those things. Okay, so you can watch those classes also. And if you have any doubt, okay, this type of if you try to solve any question like this, and you found that you are facing a difficulty, so you can send those questions, you can send those doubts to me in this comment section also. And there's another way, so there is one link in the description, so you can go through the link and you can send the question over there. Okay, so thank you so much, and make sure you like and subscribe this video, and you subscribe the channel and share this video with your friend. Okay, so thank you so much. Thank you for joining, and we'll meet in the next class. Thank you.